Okay, so today we are going to discuss about post-transcriptional processing of RNA. We all know that uh, from DNA we will get RNA, and then from RNA the required protein is produced, as the RNA contains the codons for the amino acids that are synthesized during protein synthesis or translation. Actually, DNA makes copies of itself by the process of replication, and then DNA acts as template for the making of RNA. So this process is called the translation. So in the translation, the pre-mRNA that is produced, has to be further processed to produce the mRNA. So this involves the post-transcriptional processing of RNA. So the post-transcriptional processing of RNA includes three main processes. Number one is capping, which takes place in the 5' end of the RNA, and then there is polyadenylation, which occurs in the 3' end of the pre-RNA, and also there is another process which is called splicing. What is splicing? Splicing is the process of removal of non-coding part or the introns from the pre-mRNA, and the coding parts or the exons are joined together to form the mRNA. So today we are going to confine our discussion on these three parts of the post-transcriptional modifications. Number one is capping, which occurs in the 5' site. Then number two is splicing, or removal of non-coding parts, and number three is the polyadenylation, or addition of polyadenosine residues to the 3' end of the RNA. Okay, this is the schematic representation of all the three processes. So coming to the primary transcript, you can see there are exons and introns. In this figure, we have shown three exons, exon 1, exon 2 and exon 3, and there are two introns, intron 1 and intron 2. So what happens is that in the 5' end, you can see this red color cap which is added, and if you go to the 3' end, you can see this A multiple A, or poly A tail. This is in the 3' end. What is the significance of this capping? The capping imparts stability to the mRNA, because we have seen that while we extract the RNA, it is comparatively more unstable than the DNA. Actually, what happens in the cell also, while one each nucleotide is added to the RNA, from the other side it starts degrading. So in order to impart stability to the RNA molecule, this capping is necessary, one in the 5' end, another in the 3' end. Then these introns are non-coding parts, so this has to be removed and the exons are joined together, and that makes the mRNA. So here in this figure you can see there are 3 exons, 1, exon 2, exon 3, and these spliceosomes excise introns and join the exons, and that results in the compact RNA, which is the mRNA, having exon 1, exon 2, exon 3. So this is a type of splicing which is called constitutive splicing. That means there is no alternations, just the introns are removed and exons are joined in the order which is in the primary transcript. So this is in this figure we have shown the constitutive splicing. There are other non-constitutive splicing also, which we will discuss later. So in this figure 1, this is showing constitutive splicing, 5' capping and 3' poly A tail. Now a little bit details about the capping. In eukaryotic cells, most RNA capping enzymes add a 7-methyl guanosine structure to the 5' end of the pre-mRNA by a 5' to 5' triphosphate linkage. Okay, so there is 7-methyl guanosine structure in the 5' end of the pre-mRNA, and this linkage is 5' to 5' triphosphate linkage. So this one I will show in the next figure, where it will be very clear the type of linkage. And in multicellular eukaryotes, like in human and some viruses, further modifications exist, including methylation of the 2' hydroxy groups of the first 2 ribose sugars of the 5' end of the mRNA. So here you can see the first 2 ribose sugars, the 2' position, it has methylated 2 hydroxy groups. So let us go to the figure. So you can see here that 5' to 5' triphosphate bridge. You can see here 3 phosphates are there, and that is 5' to 5', and this is the 7-methyl guanosine, 7-methyl guanosine. And if you go to the RNA chain, you can see this, the first one is having 1 methyl group attached to the second carbon, and also the second nucleotide is also having 1 methyl group. The third one is having the usual OH group. So this additional methylation also helps to protect this cap. This cap structure protects the RNA from nuclease activity. That's why this 5' cap is necessary to impart stability to the mRNA. So coming to splicing, as I have told earlier, the splicing is the process of removal of non-coding sequences or the introns from the coding sequences. So for this we need spliceosome. Spliceosome is a large and complex molecule formed of RNAs and proteins that regulate the process of RNA splicing. The spliceosome is composed of 5 small nuclear RNAs or snRNA, small nuclear RNA, and about 80 protein molecules. The combination of RNAs with those proteins result in the formation of RNA protein complex termed as small nuclear ribonucleoproteins or snRNA RNPs. Okay, so there are mostly confined within the molecules nucleus, where they remain associated with the immature pre-RNA transcripts. Okay, so all spliceosomes are involved in both removal of introns and also the ligation of remaining exons. So, as I have told earlier, there are 2 types of splicing. Constitutive splicing, that is the simple splicing. That means the order of the exons remains same and simply the introns are removed. So there is no change. But another type of splicing is the alternative splicing, where there is deviation from the constitutive splicing, and this is very important to make different types of proteins, and alternative splicing plays a very important role in like evolution. Then also we can analyze the alternative splicing for cancer variants. So let us show some figures to explain it in a very easy way. So in this figure you can see there are 5 exons depicted in different colors: exon 1, exon 2, exon 3, exon 4 and exon 5. So if it is constitutive splicing, in that case what should have happened? All the introns would have been removed and exon 1, 2, 3, 4, 5 in a sequential way should have joined to make a protein. But here you can see alternative splicing has taken place. For example, exon 1, exon 3 and exon 5 is making one protein 1. That means exon 2 and exon 4 have been removed. So this is alternative splicing, where 2 exons have been skipped. And in another one you can see where in the second one exon 1 is missing, exon 2, 3 and 4 are there, exon 5 is again missing. That also results in a truncated protein having the coding sequences for the exon 2, 3 and 4 respectively. Then another one, the last one, you can see the exon 1 is there, exon 2 is there, 3, 4 have been skipped and 5 is there. So this also making truncated protein, and we are getting 3 types of different proteins: protein 1, protein 2 and protein 3. So this is example of alternative splicing. So here you can see different types of mRNA splicing. For example, number 1 is constitutive splicing. Here we have 4 exons, so they are just joined one after another after removal of the introns. So this is constitutive splicing, number 1. Number 2 is exon skipping or exon inclusion. Exon skipping, for here exon 1, 2, 3, 4 exons are there. Here you can see that exon 3 has been skipped. So exon 1, exon 2, then exon 4. So that means it results in a truncated protein with only 3 exons. Then we have alternative 5' splice sites or we have alternative 3' splice sites. So this also results in different types of proteins. Then intron retention. What is intron retention? Normally introns are removed because they are non-coding parts. But sometimes it happens that some introns are not removed, it is also retained. So it results in a situation where some introns are remaining. Here you can see there are 3 introns. In this figure you can see the third intron is retained as a gray color. So this is the result. Or mutually exclusive exons. You can see here after 1 there is 3 and 5, or sometimes they change the position. So there are lots of permutation combinations possible, and nowadays we use NGS, next generation sequencing analysis. It has become easier to study the alternative splicing mechanisms, and also to study the variants, particularly it has lot of relevance in agriculture also, and also in medicine. So what are the applications? The pre-mRNA splicing is a fundamental process in cellular metabolism that plays an essential role in generating protein diversity. So with this splicing, we can get different permutation combinations producing different types of proteins, and the diversity is brought about by changes in the number and sequence of exons and introns present in the RNA sequence. RNA splicing also helps in regulation of gene and protein content in the cell, and splicing of RNA sequences assists in the process of evolution of new and improved proteins. Okay, and this is also very important in the medical field. Various aberrant splicing isoforms act as markers for cancers and as targets for cancer therapy. Okay, and mRNA splicing is a key to the pathology of cancers, where it regulates the 3 functional aspects of cancer, that is proliferation, metastasis, and also apoptosis. Apoptosis is programmed cell death. When something abnormal happens in the cell, then it goes for apoptosis, not the normal death, it is programmed cell death. Okay. And now coming to the last part, that is polyadenylation. Polyadenylation is the addition of poly A tail to the RNA transcript. Typically a messenger RNA, the poly A tail consists of multiple adenosine monophosphatase. In other words, it is a stretch of RNA that has only adenine bases. So you can easily recognize the 3' end by just checking for the poly A tail. So what is the function of poly A tail? It imparts stability to the RNA and prevents its degradation. Now RNA editing. What is RNA editing? RNA editing involves insertion, deletion, or modification of nucleotides post-transcriptionally. That means after transcription. The most common RNA editing in higher eukaryotes is adenosine to inosine, or A to I base modifications. Now coming to ribozymes. If you break the term ribozyme, that means ribonucleic acid enzymes. When RNA acts as a catalytically active molecule, then it is called ribozyme. Where RNA solely provides the catalytic activity. There are some examples, for example, hammerhead ribozymes, VS ribozyme, or hairpin ribozymes. Now coming to inhibitors of transcription. Okay, the transcription, from the name you can understand there are certain compounds that can inhibit transcription. For example, alpha amanitin. Alpha amanitin is found in some mushrooms. You know that it is not advisable to eat each and every mushroom because some mushrooms we call it poisonous, but actually what happens is it contains alpha amanitin. So it is a type of inhibitors of transcription. So alpha amanitin is a molecule made from death cap mushroom. The mechanism of action is that the alpha amanitin inhibits RNA polymerase II at both the initiation and elongation stages of transcription. So one single mushroom could very easily lead to fast death in 10 days. So we should be very careful before choosing the mushroom. We should use only edible mushrooms. Otherwise, the alpha amanitin can inhibit the transcription. So another one is actinomycin D. This is also inhibitor of transcription. Then rifampicin. So this one is a very different types of inhibitor where it binds to bacterial RNA polymerase but not to eukaryotic RNA polymerase. So what this rifampicin can be used is that it is a powerful drug for treatment of bacterial infections because it will not act on eukaryotic RNA polymerase, like for instance our RNA polymerase. So it will only inhibit transcription in bacteria. So it can be used to treat bacterial infections. Particularly it is used for the treatment of TB, tuberculosis and leprosy. So this is a very short description about inhibitors of transcription. So due to shortage of time, we could not go into the details. Just I gave a brief overview about the post-transcriptional modifications, and the three main post-transcriptional modifications are 5' capping, 3' poly A tail, and the splicing. It may be constitutive splicing or non-constitutive or alternative splicing. Hopefully you have gained the basic knowledge about post-transcriptional modifications, which are very much necessary for our day-to-day life activity in our cells. Thank you very much for your patient hearing. So see you with another video. Till then, goodbye.